Good afternoon and welcome. I'm Jennifer Pietenpol, one of the Chief Scientific Advisors for Susan G. Komen. This afternoon, it's my pleasure to introduce one of the two winners of the Brinker Awards for Scientific Distinction. These awards honor leading investigators for their pioneering work in breast cancer research and treatment in two categories: Scientific Distinction in Basic Science, which is being presented today, and Scientific Distinction in Clinical Research, which is being delivered tomorrow at 11:30 in this same location. The 2022 Brinker Award for Scientific Distinction in Basic Sciences is presented today to Dr. Jeffrey Wall. Dr. Wall is the Daniel and Martina Lewis Chair and Professor in the Gene Expression Laboratory at the Salk Institute, and an adjunct professor at the University of California, San Diego in the Department of Biology. He is being honored today for the significant contributions to breast cancer research, which have generated critical insights to the field of cancer genetics, including mechanisms of breast cancer drug resistance and genome stability, which he's going to tell you about today. He's a true pioneer in these studies. He's revealed many genetic drivers of breast cancer and actually revealed to all of us the immense heterogeneity of normal cells within the breast tissue. He has notably and continuously pushed breast cancer research to new frontiers by tackling the unanswerable biological questions by developing new methods and technologies and accelerating discoveries across many disciplines worldwide. Without question, Dr. Wall's scientific accomplishments have been and will continue to be paradigm shifting and leave long-lasting impressions on the breast cancer community. Congratulations, Dr. Wall, and thank you for sharing your presentation with all of us today.
Thanks very much, Jennifer. Thank you to the nominating committee for making a very difficult, I'm sure, selection. As all of the nominees, I'm sure, were equally qualified. So I'm humbled to accept this award today, which Jennifer said is kind of a career synopsis. The first thing I'd like to do is advance the slides. There we go. So this marks a truly remarkable week in my life because on Saturday we were in Sydney, Australia, celebrating the wedding of our daughter, Allison. And today, of course, I'm in San Antonio, Texas, so I have no idea of what time zone I'm in. And we'll endeavor, as Jennifer has impressed upon me many times, to finish this in the allotted time. I want to thank my family for their support over the 45 years of career that I've had at the Salk Institute, and especially my wife, Barbara Parker, who's a clinical oncologist specializing in breast cancer, who has taught me much about the clinical management and complications of these many diseases that we put under the umbrella of breast cancer. I accept this award not for myself, but instead in honor of the many trainees who I've been privileged to mentor over the past 45 years. It's their work and dedication that's led to the discoveries that Jennifer mentioned. And before I begin on the science part, I just want to give you a few career insights. And in fact, to stay on time, I'm only going to give you two. The first has to do with the top one, which is to communicate with people, especially those in different fields, because it's at the intersection of different fields that the most significant advances come. And this will be a consistent theme throughout my talk. The second one has to do with the importance of collaborating with an advocate. For all of the bench scientists in the audience, incorporate an advocate into your lab. It can be life-changing. In the case of Bianca Kennedy, who started with us 11 years ago, that is actually physically the truth, because a year after she joined the lab, she had a breast cancer recurrence. We got her to the right care at the right time and the right treatment, and now she is 10 years without detectable cancer after that diagnosis. So that's been life-changing. And in the process of her attending weekly lab meetings, she's learned about how very difficult it is to turn data into discoveries that are publishable and fundable for future research. She has been our strongest advocate locally and nationally for increased cancer research funding. The second point I want to make has to do with mentorship, and it gets to funding again. And this is, I highly urge you to read this short piece by Lan Nguyen Chaplin, who was published in Science just recently, where she talked about the importance of mentorship that enabled her to actually pursue a career in academia, coming from a family of 11 Vietnamese immigrants with no prior educational experience. She talked about mentoring with grace, which is an acronym for generosity, respect, advocacy, and be a champion of empowerment. And only in this way can we nurture, inspire, and enable the next generation. I want you all to consider our graduate students and postdocs as essential workers. What kind of experiments could you do without their effort? But they aren't paid as such. They should be. We need to advocate for changes nationally to increase the pay so that they can live comfortable lives to enable them to do the life-changing, life-saving work that they perform every day. Now I want to get to the main topic of my talk, which has to do with my career-long interest in the origins of intratumoral heterogeneity, since this clearly complicates diagnosis, prognosis, and the design of effective treatments under the rubric of precision medicine. Now, for any of you who have studied the structure of the mammary gland, you know in normal individuals that it's a very stereotyped structure. The ducts have two kinds of cells, luminal and basal, indicated by the cytokeratins here, keratin 14 in red and keratin 8 in green, and every cell has the same number of chromosomes. But in the genesis of cancer through multiple steps of evolutionary selection, especially in this case of a triple-negative breast cancer, the tissue is highly disorganized. The pathologists call it undifferentiated. The cells have different numbers of chromosomes with various numbers and types of abnormalities. And the structure of the tissue itself doesn't resemble that at all in terms of cellular phenotype to that that's present normally. Whatever cell initiated this has clearly undergone chromosomal instability and cell state plasticity. And the consequence of this is the genesis of subsets of cells that may be drug resistant, that may fuel metastasis, and that increase overall to tumor fitness. Now, over the 50 years, the black box that used to exist has been filled in summary by this nice diagram from Doug Hanahan recently, where we know that cancer cells evolve from normal cells by accumulating multiple mutations in critical steps of metabolism, immune evasion, constant proliferation, et cetera. Today, I want to talk about the interrelatedness of 4 of them that you may not think about being interrelated in others otherwise. One has to do with the genetic instability that started my career. The second has to do with the relationship of this to unlocking cellular plasticity through the epigenomic changes that are necessary for that. And finally, tissue disruption, perhaps associated with inflammation, that can cause rapid, profound cell state changes. And I'll cover this at the end of my talk. So I want to give you a story in 3 chapters. The first starting with genomic instability. If you break it, you bought it. You can't go back once it happens. How P53 prevents that from happening in normal cells, but is disabled in many cancers to unleash not only competence for many forms of genomic instability, but also for the acquisition of cell state plasticity. And finally, talking about normal and cancerous mammary cell hierarchies. It all starts with a conversation, in this case with my advisor, postdoctoral advisor, George Stark. Here's my wife, Barbara Parker. It started with 2 questions. One, who's that woman? He said, that's Barbara Parker. We ended up being married a year later. The second question changed the course of my career over the next decade, and that was the following. We had been studying mechanisms of resistance to a drug that inhibits de novo uridine biosynthesis at the same time that Bob Shimke was studying resistance to methotrexate, and we came to the same conclusion at the same time. Resistance to these inhibitors was mediated frequently by the genesis of abnormal chromosomes that contained extra copies of the genes that that were the targets of those drugs. In his case with methotrexate dihydrofolate reductase, in our case with Pala-CAD. The same kinds of abnormal structures were also being observed in cancers, in this case breast cancer, with double minute chromosomes, where the chromosomes were later shown to contain increased copies of oncogenes, such as c-Myc or one of the cyclins. But I asked him the question, well, George, we've been studying cancer cells for resistance. What happens in normal cells? And he said to me, the response of normal cells to the drug we were using was nothing. Well, it wasn't really nothing. It was that the drug caused the cells to arrest their proliferation. And that told me something very important. It said that the acquisition of genomic instability indicated by gene amplification was something that was under control, and that control was violated during cancer progression. And so for the first 10 years of my career, we searched in my lab for what those control mechanisms would be. To summarize a lot of hard work that ultimately involved the generation of new technologies to study the very earliest steps in gene amplification, we found that that early step involved the entry of cells under nucleotide limiting conditions to accumulate stalled replication forks that were prone to breakage to generate all the forms of chromosome instability that people had reported over decades in cancer cells: extrachromosomal elements, translocations, chromosomally amplified regions, aneuploidy, et cetera, and deletions. They all came from the same basic structure, intermediate structure. So then the question became, well, what is preventing this from happening in normal cells? And you can think about it in the following model. Normal cells have a way of sensing a stress, for example, a break in the chromosome, the break that initiates gene amplification. And that break activates a guardian. And the guardian prevents cell cycle progression, and this will, of course, prevent the emergence of clones with genomically unstable genomes. But in cancer, there's a mutation, or one or more, that disable this guardian, that enable the cell to proceed into the cell cycle even in the presence of chromosome damage, leading to the emergence of clones that manifest genomic instability in the ways that I said. So the question was, what is that guardian? Well, in the days before Google, I started to look through the literature for evidence of cancer cells that might have the same kinds of chromosome anomalies that we were seeing in cells that were undergoing these early stages of gene amplification. And I happened to run across this paper published in 1990 from Mike Tansky and Louise Strong at MD Anderson. And what I saw is that they were studying patients with Li-Fraumeni syndrome. And here you can see the formation of dicentric chromosomes, acentric elements, et cetera. But the gene that was responsible for Li-Fraumeni syndrome in 1990 hadn't really been clarified. But the next year it was. And that turned out to be P53. And P53 fulfilled all of the characteristics of the guardian that we were expecting. It was a tumor suppressor. It had previously been thought to be an oncogene, but work from Bert Vogelstein and other labs clearly showed that it was a tumor suppressor. Loss of heterozygosity occurred in a great majority of human cancers. Most importantly, the mutation in Li-Fraumeni syndrome was found to be a germline mutation in the P53 gene itself. And overexpression studies showed that overexpressed P53 could cause the cell to arrest in the cell cycle. We showed in G1, and later work by Tyler Jackson, our own lab, showed that it also controls progression through G2M. And very importantly for us, Mike Kasten showed, actually at the same time as we were doing this work, that P53 is activated by DNA damage to, in fact, induce that cell cycle arrest. So P53 fulfilled all of the characteristics of the guardian that we were looking for. And just the next year, armed with a strong hypothesis, Yu-Shin Yin performed the experiments that proved that loss of P53 function as Li-Fraumeni cells progressed through culture was associated with acquisition of competence for gene amplification, as shown by this summary of several years' worth of cell culture experiments. And most importantly, we fulfilled Koch's postulates by showing that if we put wild-type P53 back into a Li-Fraumeni fibroblast that had 2 copies of mutant P53, that this restored cell cycle control and it prevented the cells from undergoing amplification. That's shown in this last line here. Okay, so now we knew that p53 was the guardian of the genome. Is that all that it did? Well, for the next 10 years, I studied mechanisms by which p53 was activated, and that provided the background that I needed. Oh, I should mention that in the same issue of Cell that we published this work, Theotisty and Tyler Jacks, using mouse models, came to the same conclusion. So that prepared me for the next conversation that I had that was field changing. That was with my colleague and office mate, Juan Carlos Espisuel Belmonte, who's a developmental biologist. He had been studying cell state reprogramming, such as that was first described by Takahashi and Yamanaka, where addition of just 4 genes to normal fibroblasts could lead to their reprogramming into induced pluripotent stem cells. And I asked him a simple question. I said, this is really a remarkable process, but why does it occur at such a low frequency? And he said, I really have no idea, but if we could get it to occur at a higher frequency, that would increase the probability and the utility of this process for a variety of forms of medicine. And I said, well, from my experience with p53 work, I would bet that the oncogenes that are used here, such as c-MYC, were activating p53 to prevent the cells from going through the cycle to generate the clones that would be reprogrammed. Again, armed with a strong hypothesis, we set forth a series of very straightforward experiments to inactivate p53 or to overexpress it, to activate it by drugs such as Nutlin, to ask whether inactivating it increased the frequency of reprogramming it, and activating it prevented such reprogramming from occurring. And we published those results in 2009, collaborative work in Nature. Other people had the same idea. They did very similar experiments using different cell sources and different strategies, and 5 papers were published simultaneously in the same issue of Nature, coming to the same conclusion, convincing us that our interpretation was correct. This led Scott Lowe to write a review of these papers in which he said, are these results a heads up that cancer cells and stem cells are disturbingly similar? I presented our work at a p53 meeting where I had another conversation with one of the leaders of that field, Arnie Levine. Arnie and I had the same idea. We said, well, in cancers that have frequent p53 mutations, perhaps the cancer cells were reprogramming to a primitive state. And so we looked at the breast cancer collection from TCGA as it had been annotated so carefully, and we asked whether there was a correlation between acquisition of these signatures that were indicative of embryonic stem cells or induced pluripotent cells, and correlation with either p53 mutation or mutations that were predicted to inactivate p53. And you can see that in breast cancers, the highly differentiated ones have no such enrichment for this signature. The ones that are least differentiated, such as the basal-like, do, and these are the ones that most frequently either initiate with p53 mutation or have other mutations that disable p53 function. So this led to the following idea, that just as in vitro, in vivo, p53 might act as a guardian against reprogramming to a primitive state by inducing apoptosis or cell cycle arrest or enforced differentiation in order to prevent cells from reprogramming. But on the other hand, when p53 function is abrogated, then what happens is the cells can de-differentiate. And the question became, what state are they de-differentiating to? We didn't think it was like an induced pluripotent state or an embryonic stem state, but rather it might be something that was suggested as early as the 1800s, where the Italian surgeon Francesco Durante, working with Cohnheim and Virchow, suggested that cancer cells subjected to persistent inflammation could actually change their state. He said to a very primitive state, I'm inferring an embryonic state here, where the cells could differentiate into the multiple cell types that he was observing in cancer just by use of a microscope to generate intratumoral heterogeneity. So we started to look at mammary development to ask whether there was any relationship between cells formed in early mammogenesis and those that we were seeing in cancers where we knew reprogramming was occurring. And you can see in this analysis of mouse mammary development starting in relatively late embryogenesis, embryonic day E18, the cells all exhibit kind of a mixed phenotype, where each cell has a mixture of both basal and luminal characteristics. Soon after birth, you start to get specification of the basal layer, but no frank lumen. And in the adult, you can see the stereotyped ductal structure with beautiful differentiated myoepithelial or basal cells on the outside, luminal cells on the inside. If you look at a human breast cancer and you use the same markers for evidence of states of differentiation, what you see is something actually in this region that looks remarkably similar to what we see in the embryonic mouse. There are cells that appear to be in a hybrid state of differentiation, others that seem to either be myoepithelial or luminal, and some that are neither, even though all of these are epithelial cells. So the cell that initiated this tumor has obviously undergone dramatic cell state plasticity. So the question was, to what state are they reverting? And so that started our analysis of, at the single cell level, eventually, of mouse development to understand whether there was a relationship between cells formed in the embryo that are that may be the bipotent cells of the mammary gland, and those that we see in the most aggressive basal-like breast cancers. And that paper was published in 2012 by the authors shown here. And we had analyzed these cells functionally using transplantation and in vitro using mammospheres, where a single one of these embryonic cells would form a mammosphere that had both basal and luminal cells, and if given lactogenic hormones would make milk, which was remarkable. But at the same time, work from Cedric Blancpain's group using fate mapping came to the same conclusion, that it was cells in the embryo that were bipotential, and that after birth, these cells became unipotential in their abilities. In other words, luminal cells generate luminal cells, basal cells generate basal cells. This was in disagreement with the literature at that time, and I'll come to a resolution of that disagreement at the end of my talk. I just want to go through a few of the characteristics of these cells that we analyzed transcriptomically. Their transcriptomes of these fetal mammary stem cells, we call them, were most similar to that seen in basal-like breast cancers. By contrast, the kinds of cancers that have the highest concentration of what have been called cancer stem cells most resembled what we now know to be the cells that Jane Visvater was calling adult mammary stem cells, which are really basal cells of the adult mammary gland. They also resemble the stroma that we find in the fetus. So if you think about stem cells in basal-like breast cancer, they really represent, or most closely transcriptomically resemble, this fetal state. They don't at all resemble what have been called cancer stem cells, which are really mesenchymalized variants of an epithelial cell. The final thing is that the signature that we derived is very different from any of the other signatures that have been presented up to that time. That's reasonable because were studying that state in the embryo that had never been studied before, and we found that this was of prognostic significance even in multivariate analysis. Then we knew at that time that these cells were highly heterogeneous. Even though they were functionally similar, transcriptionally they were very heterogeneous. And we did that by the first primitive single cell analyses where we could just analyze 96 of these cells with 96 genes. That showed that they were very different from one another. So then we started to apply the most advanced methods of single cell biology available at the time, developing the first single cell transcriptomic and epigenetic analyses of mammary development starting from the embryo through the adult. And these heroic postdocs were the ones who conducted that work. I'm just going to summarize it, 10 years worth of work in the following way. First, the stem cell state is really a mixture. Each cell is a hybrid of both basal and luminal characteristics. It's a mixed luminal basal state. The cells are all different from one another, even though they manifest functionally in the same way. So there's a lot of heterogeneity that's reasonable in terms, if you think about these cells are in different microenvironments. Differentiation occurs gradually, not suddenly. And the cells that are formed after differentiation are transcriptionally and epigenetically very heterogeneous from one another. All of this data powered us to understand the factors that were involved in differentiation, including the identification of a number of SOX factors, including SOX 9 and 10. SOX 10 is an extremely interesting one because it's essential for the fetal mammary stem state. We knocked it out, Chris Dravis did, those cells were no longer functional. And when overexpressed, even normal cells would transform into a neural crest-like state that was highly aggressive and invasive. And this neural crest state we observed to be present in triple-negative breast cancers. And because SOX10 is a transcription factor and has many downstream targets, many actionable targets were elicited by this. And I urge you to, especially the clinicians in the audience, to go back to that paper, because I think you'll find some new targets for triple-negative breast cancer. This is the classic view that's been generated that you've heard about before, about mammary development. Instead of thinking about this kind of very discrete view of mammary stem cells going into defined cell types identified by just a few markers each, rather, I think a more accurate view based on our analyses as well as those from many other labs at the single cell level is that in the embryo, bipotent cells arise, probably very soon after the skin starts to differentiate into the mammary gland. These cells are bipotent, but after birth they specify into unipotent progenitors that generate the luminal lineage and the basal lineage. But within both of these lineages, the cells are very heterogeneous, but they're conditionally plastic. And what I mean by that is that if you, for example, disable tumor suppressors such as BRCA1 or P53 in mature luminal cells, as was done by Walid Khalid's lab, these cells that are in a virgin female will undergo alveogenic mimicry. They'll produce milk. Similarly, if you introduce activating mutations in PI3K, either in basal cells or in luminal cells, these cells can transfade. Basal can turn into luminal, luminal can turn into basal, and they can generate different types of cancers. And in work that was just recently published from Daniel Schrammick's lab that was actually promoted by a discussion that we had at the San Antonio Breast Cancer Conference in 2019, he showed that mutations in the long tail of genes, 215 of them, many 40% of which are comprised in the BAP1 and COMPASS epigenetic regulators. These, in combination with PI3K mutations, even in basal cells, will cause the basal cells to undergo alveogenic mimicry and form what appear to be luminal-like tumors. That still raises the question, what about these basal cells that by the elegant work of Connie Eves and Jane Visvater were interpreted to be adult mammary stem cells by transplantation studies? Well, work by Cedric Blampain and many others by lineage tracing showed that these are really unipotent. Basal cells generate basal cells in the context of a normal gland. So how can you resolve this difference? And we realized that a big difference between the strategies used for analysis had to do with tissue damage. In transplantation, of course, you disrupt the tissue, you take individual cells and transplant them into a mouse in which all the epithelium has been removed. Is it possible that these basal cells, when freed up of their local constraints, can rapidly reprogram into both lineages that form the mammary gland and in that way regenerate it? So we generated a whole new mouse model to be able to study this process in real time. We used CRISPR to knock into the keratin 14 locus and in the keratin 18 locus different colors. Keratin 14 green color, keratin 18 a red color, so that every cell in this gland, if it underwent a state change, could be revealed by a difference in its color. And so Nikki Lytle did a heroic experiment where she injected 10,000 cells per time point into the cleared fat pads of either neuter immune competent mice, of pure basal cells isolated because they were green, and over time studied what happened to them. And within 4 days, you can see that the cells that were transplanted rapidly changed their fate into a hybrid basal luminal state, which then rapidly progressed in differentiation until by 8 weeks you regenerate the entire mammary gland that was used prior to transplant. And so then the question becomes, well, is this just an example of what happens under extreme tissue disruption, such as you see in transplantation experiments? And the answer is no, because Cedric Blancpain, again, did a beautiful experiment based on the original work of Pietenpol, where he selectively ablated luminal cells, and that led to basal cell reprogramming. But he didn't understand mechanism, and he didn't understand rate. Chemotherapy, by the way, by the work of Ian Macra, can do the same thing. So what is the nature of what we call these developmentally unstable hybrid cell states, or DUCs? So we did the first multiome analysis over time after transplantation, meaning that from individual cells, we isolated single cells and analyzed both their epigenome and their transcriptome to ask about mechanisms by which this was happening. And what occurs very rapidly is the basal cells used for transplantation go through a fetal-like state that is bipotent, whereupon they can differentiate into both mature basal cells, luminal, what are called luminal progenitors, as well as mature luminal. We know the cells are going through a fetal state because they express SOX11, which is only expressed in the fetus or soon after birth. And interestingly, in all models of mouse mammary cancer that mimic a triple negative state, we see the expression of SOX11, meaning that these mature adult cells have reprogrammed to an embryonic-like state. And in addition, using published single-cell data from humans, from BRCA1 carriers that have been untreated, we see that many of these cancers also express SOX11, meaning that in humans this kind of reprogramming is also going on. I just want to focus on SOX11 for a second, and I'm going to finish in a minute. SOX11 has been widely implicated in tissue repair and also in breast cancer, first by elegant work from B. Howard's lab, later from Powell Brown. So it's a gene to reckon with, and we're looking at its functional importance now. And I just want to finish with this summary slide, where in normal tissue injury, these developmentally unstable hybrid cell states are generated, but they're rapidly resolved to heal the wound. This is something that occurs in many different tissues. It's not just in breast cancer. But in the presence of a continuous genomic insult, such as you would get in a BRCA carrier, you get a persistent inflammatory state that causes these cells to stay around. Of course, they have oncogenic mutations in them where they can initiate cancer. And so I think that there are a number of mechanisms for this, and there are many therapeutic opportunities. One of the mechanisms involved DNA damage-induced activation of the CGAS STING pathway, as has been reported to occur in cancers with BRCA phenotypes. But another mechanism involves disabling the P53 and RB pathways, activation of JAK-STAT and FGF, as occurs in prostate cancer. Given the similarities of prostate and breast, I'm pretty sure that a similar thing happens in breast when we look for it. So with that, I'd like to finish. I want to honor all the people who have worked with me over the years, all of the funding agencies who have supported this work that at many times has been very high risk, and the people who are currently in the lab, the wallabies, who thank you for your attention and your support.